Hello everyone, Ms. Duco again. This is video number 2 of the Read Aloud articles. I am now going to be reading Science News for Students article. Okay, this one is titled Molecular Scissors Fix Disease-Causing Flaw in Human Embryos. Again, highlighting things that you think could be useful for your argument. Okay. Researchers used molecular scissors to mend a faulty gene in human embryos. These tests are the first of their kind in the United States. Scientists used an enzyme known as CRISPR, or Cas9, as their scissors. They snipped out the faulty gene, which can lead to heart failure. With this tool, they fixed the problem in nearly 3 out of 4 treated embryos. That's about 72%. Such tinkering with an embryo's DNA is known as germline editing, and it is very controversial. Many people worry that it could be used to create so-called designer babies. Parents of these still fictional designer babies might try to choose their child's eye and hair color, or they might have doctors change certain genes to make their child into a super smart athlete. In fact, the new study suggests that designer babies might be very hard to make. They would require injecting bits of new DNA to supply the desired traits. And in the latest test, treated embryos rejected pieces of the DNA that the researchers had added, in this case to help fix the faulty gene. So it's not guaranteed that the genes that they introduce will be accepted by the body. In this case, some of them were rejected. The new work is not announcing the dawn of the designer baby era, says R. Alta Charo. She is a lawyer at the University of Wisconsin Law School in Madison. She also is a bioethicist, someone who studies the wisdom, morality, or social value of certain decisions or policies. The researchers have not attempted to add any new genes or change traits, she notes. Here they sought only to repair one version of a disease-causing gene. This research offers lessons in how to more accurately wield the CRISPR scissors. They reduce the share of embryos that were only partly edited. That's important if researchers want to use these gene scissors to cure genetic diseases, says Sharat Matapov. He is a co-author of the new study. A reproductive biologist, he works at Oregon Health and Science University in Portland. If even one cell in an early embryo is unedited, he explains, that's going to screw up the whole process. His team did work in Korea, China, and 2 United States, Oregon and California. They described their achievements on August 2nd in an article called Nature. Snip, then repair. Mutations are changes that occur in a gene. Some mutations lead to disease. Others seem to cause no harm at all. In the new work, researchers wanted to fix a mutation in the gene known as MYBP3C. Nearly every cell of the body has 2 copies of each gene. People who inherit just one mutant copy of MYBPC3 can develop a life-threatening heart problem. It's called hypertrophic cardiomyopathy. It can cause sudden death from heart failure. The heart problem affects about 1 in every 500 people worldwide. Mutations in the MYBPC3 gene are to blame for about 40% of these cases. Doctors can treat symptoms of the condition, but there is ultimately no cure. In the new study, researchers used sperm from a man who carried a flaw in the MYBPC3 gene. They injected that sperm into eggs from women with healthy copies of the gene. The researchers also injected the gene editor into the egg. The editor has several pieces. One is the scissors, a DNA cutting enzyme called Cas9. They also need a piece of RNA that directs the enzyme to the exact place in the gene where a cut should be made. The researchers injected another piece of DNA into the egg too. The researchers had designed that hunk of DNA to serve as directions for repairing the breach that would be made by Cas9. but the cells ignored it. Embryos instead used the mother's healthy copy of the gene to mend the cut. The results surprised the researchers. Gene editing in other types of cells usually requires an added piece of DNA as a pattern to guide a repair. If all embryos reject added DNA repair patterns, researchers may struggle to fix mutations in embryos. It would be hardest when neither parent has a healthy copy of the gene. The finding also means that embryos may reject attempts to add new traits. That might mean designer babies would be hard to achieve. Okay. Careful timing. Timing is important in these experiments. At first, the researchers added the scissors one day after fertilizing the eggs. Those experiments often produced patchwork embryos, ones having some unedited cells. 13 out of 54 treated embryos had some unrepaired cells. Affected eggs that were fertilized had probably copied the DNA before researchers added Cas9. When researchers injected the cutting enzyme in sperm at the same time, this did not occur, and none of the resulting embryos showed any signs that Cas9 had snipped a cut in the wrong place. One concern about gene editing is that off-target cutting could create errors in other parts of the cell's DNA, which could result in new disorders that we don't know about yet. When an egg was treated at fertilization this way, only one patchwork embryo occurred. It had repaired the MYBPC3 mutation in all of its cells. Some cells had used the mother's DNA for repair. Other cells used the DNA template supplied by the researchers. The new study is a step toward using gene editing to prevent genetic diseases, says Jeanette Rawson. She is a developmental biologist in Canada at the Hospital for Sick Children in the University of Toronto. Still, she warns, researchers have a lot more work to do before they can do gene editing in humans. Doctors will need to be sure the technique will work well and every single time. Researchers have edited human embryos before, but were merely probing the early stages of human development. They also wanted to learn things about basic human biology. Mitopiv and his colleagues instead wanted to probe the use of gene editing in medicine. They want to improve the technique so that it would be safe and reliable enough to use in humans. Such studies might one day involve editing embryos in the lab, then implanting these into a woman to establish a pregnancy. In the United States, such human trials are not allowed at this time. The Food and Drug Administration will not allow research meant to introduce change in embryos that could be passed down to someone's future children and grandchildren. That is the end of this article.